you guys super confused about why the vestibulo-ocular reflex works? What's up everyone, my name is Austin Miller, I am the NeuroNerd PT, and today I'm going to talk about the physiology behind the vestibulo-ocular reflex. So in the last few months, my vestibular caseload has really been expanding because I'm trying to move more into a vestibular and concussion rehab specialization. Because of that, I've been talking a lot with some students about how to incorporate vestibularly challenging treatment options into our general balance patients, so that way we can try to improve balance, dizziness, and all of that stuff. So I actually got this image, like I drew the whole thing, but I got this image from a guy on YouTube called the Ninja Nerd. If you guys haven't seen his videos, he does a phenomenal job talking about the nervous system, special sensory systems, and just generally how the body works. And so I would highly recommend going and looking up the Ninja Nerd on YouTube. So let me give you a quick rundown of everything that is on this image. And so obviously you have the eyes. Here you have the brain stem. You've got, this is going to be representative of the left, so if I'm facing this way, the left ampulla of the horizontal canal in the inner ear labyrinth, and this is the right ampulla of the horizontal canal of the inner ear labyrinth. These are just magnified images, poorly drawn. They look like a foam finger of the cilia that deflect underneath the cupula that give our brain the sense of movement. And so let's quickly talk about what we're looking at in this ampulla. So the red portion is the cupula. Deflection of the cupula is what moves the hair cells that gives our brain perception that rotational movement is happening, or rotational acceleration. The taller little finger here, that is called the kinocilia, or sorry, the kinocilium, singular, kinocilia, plural. And that is the one that essentially dictates whether the semicircular canal is inhibited or excited based on which direction the stereocilia, which are the smaller ones, which directions they deflect, either toward the kinocilia or away from the kinocilium. And so let's talk about what is happening in this picture. So in this picture, I'm depicting what happens within the horizontal canal and how the horizontal canal talks to your brainstem and thus talks to your eyes to allow the VOR to function properly. And so in this picture, we're presuming that we just quickly turned our head to the right, okay? So what happens is you turn your head to the right, and we will start with the side that is excited. And so what happens is you turn your head to the right, and you've got this whole area filled with endolymph, okay? The endolymph rushes past the cupula, deflecting it what we call ampulopedally. Ampulopedal means that the cupula is deflected in toward the ampulla. In the horizontal canal in particular, ampulopedal movement is excitatory. Now in the posterior and the anterior canal, ampulopedal movement is inhibitory, but in the horizontal canal, it's excitatory. And so you get this rush of fluid that deflects the cupula ampulopedally, and that causes the stereocilia to deflect toward the kinocilium, okay? So let's expand that and look at what it looks like on a larger scale. So you get rush of endolymph, deflects the cupula ampulopedally, and you get the kinocilium, which is deflecting further away, and that stretches these bands to separate the stereocilia, and then these little red cylinders are the ion channels. So when this deflects toward the kinocilium, these ion channels open, and you have all these calcium and potassium ions that rush into the macula, which is what's underneath the cilia, okay? When you get all of these positive ions that rush in, you get a ton of positive charge, which leads to what's called depolarization. Inside of the macula, you have these vesicles here that stimulate the release of glutamate, but only if the vesicle channels open. When this depolarizes, you get these calcium and potassium ions that open up these vesicle gates that releases glutamate into the synaptic cleft in between the the macula, and the sensory receptor. So what happens is glutamate itself is an excitatory neurotransmitter, right? And so you get all of these action potentials, all of this positive action potentials that move toward the vestibular ganglion. And so what happens here is you get all of this excitation moving up the afferent fibers, and it goes all the way up to the vestibular nucleus in the brainstem. So let's talk about what's happening on the side that's inhibited. So we turned our head to the right side was excited, the left side, what happens is you get endolymph movement away from the ampulla. We call that ampulofugal movement. So the cupula deflects ampulofugally, and you get deflection of the stereocilia away from the kinocilium. And so let's look at what happens on a bigger level. So the kinocilium deflects into the other stereocilia, and then you get the closing of those ion channels. They do not open up. So all of this calcium and potassium is trying to go through the ion channels, but they're closed, so they deflect off, and then you get relatively negative charge in this, and so that's what causes what's called hyperpolarization. So when this is hyperpolarized, the vesicles will not release anything to stimulate the release of glutamate. So you have no glutamate in the synaptic cleft, and so that means that you have decreased amounts of action potentials leading to the vestibular ganglion. Okay, so from the right side, you have a lot of action potentials going up to the vestibular nucleus in the brainstem. On the left side, you have little to no action potentials stimulating the vestibular nucleus on the left side of your brainstem. Okay, so from there, what happens is you've got 4 different areas of the vestibular nuclei. You've got superior, inferior, lateral, and medial. And most of what we're going to focus on is the medial portion of the vestibular nucleus. And so these will actually cross over each other inside of the brainstem. And so let's look at the excited portion first. So the medial portion of the vestibular nucleus is going to send excitatory signals to this little green dot. This green dot is actually the nucleus of the 6th cranial nerve, or the abducens nerve. Okay, so when that gets excited, it's going to do 2 things. It is going to send excitatory signals all the way over to lateral rectus, because that's what abducens innervates, right? Lateral rectus. And it's going to tell lateral rectus to fire. The other thing it's going to do is it's going to send excitatory signals to the nucleus of the 3rd cranial nerve, or the oculomotor nerve. Now, the oculomotor nerve innervates several of these muscles around the eyes, but in particular, with a rightward head movement, it's going to send excitatory signals to the medial rectus in particular. Now, let's look at what's happening on the inhibited side. You're getting very few action potentials communicating with the vestibular nucleus, and so because this is being inhibited, it then crosses over, communicates or articulates with the 6th cranial nerve nucleus to send inhibitory signals. So that way, what happens is you get this efferent nerve fiber going to the lateral rectus, telling the lateral rectus to not fire or to be inhibited. And it also crosses over to talk to the 3rd cranial nerve nucleus to shut off or inhibit the right medial rectus. So basically what you get is firing of medial on the right, firing of lateral on the left, inhibition of these 2, so there's no tone that these 2 muscles have to pull against to be able to move the eyes to the left. So what is happening is the head turns right, all of this sequence is happening, and your eyes relative to your head move left, so that way you can keep your gaze fixed on the object that you're looking at, okay? So that is a relatively quick rundown on how the vestibulo-ocular reflex typically functions in our brains. If you guys have any questions about this, or you want to learn more about the vestibular system, like and leave a comment, and I'll be happy to get back to you.